Assalamualaikum and have a good day everyone. So we are from group 3. We'll present about our SEM project in this video. So before we start, let me give some introduction first. So in the field of bond tissue engineering, developing materials that closely mimic the structure of and a function of natural bond is crucial for a successful regeneration. This project focused on the fabrication of a nanofiber scaffold composed of PCL, PVP and beta TCP, a combination designed to merge mechanical strength, biocompatibility and bioactivity. So the method that we will use is by electrospinning, which we produce a nanofiber with a porous interconnect structure that stimulates the ECM. So the resulting polymer ceramic nanocomposite hold a promise as a biodegradable scaffold for bond repair and generation. So in this video, we will have 4 contents, which is types of materials, fabrication method, lists of analysis that can be done of the sample, and the discussion or remark for our SEM image.
Our group material is actually a combination of 3 things, which are PCL, PVP, and beta TCP nanofibers. So first, PCL is a biodegradable polymer, which basically means it's a kind of plastic that can break down in the body over time. This one gives the scaffold strength and kinda help it maintain its shape. Then we also have PVP, which is hydrophilic, so it absorbs water more easily. This helps to improve the porosity of the scaffold, so like cells and nutrients can move through better. And finally, we have beta TCP, which is a ceramic material. It mimics natural bone mineral and helps with bone regeneration. So if you imagine a broken bone, this beta TCP helps encourage the cell to grow new bone. So when we combine all 3 of them, it becomes a composite material that has both the flexibility of polymer and the bioactivity of ceramics. And we use electrospinning to fabricate it. And as you can see in this figure here, this method helps us produce nanofibers that are super small, like really tiny, and very porous. These fibers actually kinda copy the natural structure of bone, which is good for mimicking the bone extracellular matrix structure. So to summarize, this composite material is really suitable for bone scaffold applications because it's strong, porous, biodegradable, and also helps bone cell grow.
What is electrospinning? According to Badwaj and Kundu in 2010, electrospinning is a technique used to produce nanofiber by applying a high voltage electric field to a polymer solution. It creates fibrous scaffold that closely mimics the structure of the extracellular matrix or ECM. These scaffolds are especially useful in biomedical application because they support cell attachment and growth, as explained by Lee and Jia in 2004. One major advantage of electrospinning is that we can control fiber properties like diameter, porosity, and alignment by adjusting factors such as solution concentration, voltage, and distance between the needle and the collector. This was highlighted in the study by Palm, Sharma, and Mikos in 2006. Because of this benefit, electrospinning has become widely used in tissues engineering, as particularly for developing bone scaffold. To prepare nanofiber, we create a homogeneous solution by mixing PCL, PVP, and beta-tricalcium phosphate in a suitable solvent, commonly dichloromethane or dimethylformamide. Next, we load this polymer mixture into a syringe fitted with a metal needle. A high voltage, usually between 10 to 25 kilovolts, is then applied between the needle tips and the grounded collector. This electric field causes the solution to be ejected as a fine jet. As the solvent evaporates mid-air, the polymer solidifies and forms nanofiber. These fibers are collected on the grounded surface, resulting in a non-woven fibrous mat, ready to be used as a scaffold for biomedical application.
Next, we move to the next part, that is a list of analysis that can be done for the sample. The first one, we have TEM, or we call it as transmission electron microscopy. The principle of this one is a high energy electron beam passes through an ultra thin sample to form high resolution image of internal structure. The purpose of this one is to observe the internal morphology of electrospun PCL or PBP nanofibers, to confirm the presence and distribution of embedded beta tricalcium phosphate nanoparticle within the fibers. The key observation for TEM is TEM images reveal uniform nanofibers structures. Dark spherical beta TCP particles were clearly visible inside or attached to the fiber matrix, confirming good dispersion. Advantage for TEM is very high spatial resolution, about 1 to 2 nanometer, enable visualization of nanoparticle fiber interaction. The limitation of TEM is requires complex and ultra-thin sample preparation. And lastly, equipment is costly and requires skilled operation. These are the example of TEM images. Next, we have AFM, or atomic force microscopy. The principle for AFM is it maps the surface topography by scanning a sharp tip over the sample surface. Deflection of the cantilever are recorded to produce a 3D surface profile at the nanoscale. The purpose of AFM is to analyze the surface roughness of electrospun nanofibers. Also, to investigate surface texture and uniformity, which influence cell adhesion in tissue engineering application. The key observation AFM is analysis show interconnected continuous fiber network with moderate surface roughness. Roughness average is about 35 to 70 nanometer depending on beta TCP content. The advantage of AFM is provide 3D surface imaging with nanometer resolution. It can quantify roughness and surface features. The limitation for AFM is small scan area, about 1 to 10 micrometer square. Relatively slow scanning process. These are the example of AFM images. The last part is FTIR. The principle for FTIR is, before that, FTIR stands for Fourier Transform Infrared Spectroscopy. The principle for this one is it identifies chemical bond by measuring the absorption of infrared light as it interacts with molecular vibration, producing a spectrum of characteristic peaks. The purpose of FTIR is to confirm the presence of PCL, PBP, and beta TCP in the nanofiber matrix. Next, to verify successful incorporation and interaction of the composite components. These are the example of FTIR images. Next, we have the key observation component. For PCL, we have functional group that is C double bond O stretching, CH2 stretching. For PBP, C double bond O and CN stretching. And lastly, beta TCP, we have a PO4 3- vibration. The advantage for FTIR is quick, non-destructive chemical analysis. Next, identify functional group and polymer blending. And lastly, limitation for this one is cannot provide spatial distribution. Overlapping peaks may require further interpretation.
Okay, this is the SEM image at 1000 magnification with a scale bar of 100 micrometers. At this magnification, we can see the overall structure of the nanofiber map. While the fibers are visible, they are not clearly resolved. This image is useful for observing the microscopic distribution, coverage, and general uniformity of the mat surface. However, individual fibers and fine structural details cannot be easily distinguished. Moving on to 2500 magnification with a scale bar of 30 micrometers, the image shows a clearer view of the nanofibers. Here, we can distinguish individual fibers including variation in diameter and orientation.
The fibers appear to be randomly aligned and the interfiber porosity is more obvious. There are no visible defects like beads or breakage, which suggest stable processing conditions. Finally, at 5000 magnification with a 20 micrometer scale, we see the highest level of detail. The nanofibers are well defined with smooth surface and distinct boundaries. We can clearly observe fiber junction, overlapping areas, and pore structures. This image is particularly useful for measuring fiber diameters and analyzing microstructural features. In summary, as the magnification increase, the resolution of the fiber morphology improve. At 1000, we get a general overview. At 2500, we can observe individual fiber networks. At 5000, we get precise morphological details for quantitative analysis. This SEM image confirms that the nanofiber mat has a uniform, bead-free structure with random fiber orientation and good porosity, making it suitable for applications such as filtration or biomedical scaffolds.